Okay, let's take a look here at eukaryotic cell structure. Um, as we talked about before, eukaryotic cells contain membrane-bound nuclei along with membrane-bound organelles. This on average makes them larger and also more complex than prokaryotic cells. Going back to your general biology discussions, you should keep in mind cells make up tissues, and then the various tissues can be used to make up or compose an organ. Eukaryotic cells, as it says here, are highly variable. The major two differentiating characteristics are animal versus plant cells, although you can also have accompanying differences with the fungal cells. The animal cells have no cell wall. Both fungal and plant cells have cell walls, but they're composed of different polysaccharides. The fungal cell walls contain a polysaccharide called chitin, while plant cells contain a cell wall composed of cellulose. Table 3.6 gives you a rundown of the typical eukaryotic cell structures. Many of these you've seen before, and for the purposes of your exam, you should basically know the major functions of these various organelles. For instance, the cell membrane, which we'll discuss very briefly, is a selectively permeable barrier between the external environment and the internal environment. Cilia, short hair-like appendages used for movement. The cytoskeleton provides structural support to the cell. Flagella, longer, more whip-like appendages used for movement. The ribosomes used for protein production. In the case of the eukaryotic cells, we'll see that there's a difference between those and the prokaryotic cells. Chloroplasts used to accomplish photosynthesis. The endoplasmic reticulum used to synthesize certain molecules. Rough ER used to produce proteins, typically smooth ER involved in lipid synthesis. The Golgi apparatus, basically used for packaging of macromolecules once they're produced. Lysosomes, are digestive organelles. Mitochondria, the powerhouses of the cells used to produce ATP aerobically. The nucleus is the area where the genetic information, the DNA, is housed. Peroxisome, also used for digestion, but these are going to be used to produce hydrogen peroxide and some other potential oxygen byproducts that are typically degraded. This table 3.7 gives you a typical rundown of prokaryotic versus eukaryotic cell structure. As we've gone through these discussions, you should hopefully be aware of some of those. I'll highlight the presence or absence of a nucleus, the difference in ribosomes, the difference in the presence or absence of membrane-bound organelles. There's a difference in flagellar structure. As I mentioned to you in the prokaryotic discussion, the flagella of prokaryotes are made up of a protein called flagellin, and they move in a propeller-like movement. You'll see in this discussion that the flagella of eukaryotes are made of tubulin and move in a whip-like type pattern. The chromosome, the genetic information being different, a single circular DNA chromosome in the prokaryotic cell, whereas you have multiple linear DNA molecules within the eukaryotes. The cell wall difference, especially in structure, peptidoglycan being unique to bacteria. You got chitin and cellulose for the eukaryotic cell wall structures. The cell membrane, slightly different. So I've highlighted for you a few differences, so make sure you go back and you try to appreciate those and have a comparison of prokaryotic versus eukaryotic cell structures. We'll continue on. This is the table that continues that, but I've highlighted the major ones that I'd like you to be responsible for. So the cell membrane of eukaryotes, it's what you've always heard of dating back to general biology. It consists of a phospholipid bilayer with various proteins within it. You have some proteins function as receptors. They can bind specific molecules. If they do so, it's called a ligand, and they can be very important in cell communication. You'll see various sterols located within the membranes of these eukaryotic cells. Remember, the only prokaryotic cell that we really found these sterols within belonged to the genus Mycoplasma. The ribosomes are the site of protein synthesis within eukaryotic cells. However, unlike prokaryotic cells, the size of these ribosomes will be different. Eukaryotic cells have 80S ribosomes, whereas prokaryotic cells have 70S ribosomes. So once again, please appreciate the difference in the ribosomes. Cytoskeleton, they're to support the size and the structure of the cell, made up of a variety of different proteins. You'll have actin, which are the thinnest of those filaments. They make up structures called microfilaments. Next up, microtubules. They're much thicker. They're typically going to be found supporting the strengthening components to that cytoskeleton, but they can also be used to form flagella. And then you have intermediate filaments, which provide the mechanical support. Flagella and cilia are both structures used for motility by eukaryotic cells. Keeping in mind, both of these are composed of microtubules and therefore composed of a protein called tubulin. Those microtubules are arranged in a 9+2 arrangement. I don't usually go too far in depth into the arrangement or the exact structure and how these eukaryotic flagella work, because in most cases you've already seen that in the general biology course. So basically keep in mind what the flagella and cilia are used for and the fact that they're made up of tubulin, and you should be okay for my course. The nucleus of a eukaryotic cell houses the genetic information, the DNA. Within the nucleus, you'll see a darker area called the nucleolus, and it's the nucleolus which is where ribosomal RNA is actually produced. Nucleopores in that nuclear envelope, which is a bilayer that surrounds the nucleus. The nucleopores allow materials to move in and out of that nucleus. Mitochondria are the powerhouses of eukaryotic cells. They're where the ATP would be produced. Those mitochondria produce the ATP under aerobic conditions. It's important to note that the mitochondria have their own DNA. The mitochondrial DNA is 70S like prokaryotic cells. So, as we'll talk about soon enough, it's thought that these mitochondria originated as separate individual organisms and eventually came into a symbiotic relationship with the larger eukaryotic cells. However, they retain their own DNA. So mitochondria, you should know their function and also the fact that they do have their own DNA and 70S ribosomes. Next up, chloroplasts. Similar to mitochondria, the chloroplasts contain their own DNA along with 70S ribosomes. I think it goes without saying, you should absolutely keep in mind what they're used for, and that is they carry out photosynthesis. We're not going to go obviously into all the details of photosynthesis, but you should know that chloroplasts are photosynthetic organelles. You can see up here they're only found in plant and also algae cells. This is a slide which discusses with you briefly the endosymbiotic theory. You can see up here it's simply a theory that implies that the ancestors of mitochondria and chloroplasts were their own individual cells, i.e. bacteria, and at some point they decided to join with larger eukaryotic cells in a beneficial relationship for both parties. That's the whole concept of symbiosis. Both cells exist in a situation in which it's beneficial for both cell types. For instance, chloroplasts and mitochondria allow the eukaryotic cells to produce energy, while the eukaryotic cells provide protection for the smaller, previously, so to speak, mitochondria and chloroplasts. You've got multiple lines of evidence for supporting this theory. I won't read these back to you, but suffice it to say the theory, I just summarized the theory for you. The endoplasmic reticulum, a series of flattened sacs seen within the cell used to produce in the synthesis of certain molecules. If the ER contains ribosomes, it's called rough ER, used in protein synthesis. If it does not contain ribosomes, it's called smooth ER, and that's typically used in lipid synthesis. The Golgi apparatus packages molecules so that they are able to be transported to different areas of the cell after they've been produced. And then you have both lysosomes and peroxisomes. The lysosomes contain digestive enzymes that will degrade proteinaceous components. They, as we talked about previously, are very effective against penicillin, so they would be highly effective against gram-positive cells. The peroxisomes are used to basically break down the byproducts of oxygen metabolism, that being hydrogen peroxide and something else called superoxide. These two chemicals would be toxic to a cell if you were not able to properly deal with them. And that's going to end the presentation regarding eukaryotic cell structure.